Alright, let's take a deep dive inside your cells, right into what you call the microscopic control center. We are going to follow a single set of instructions from a gene as it gets a complete makeover, and I mean complete makeover, preparing for its critical mission in the cell. Here's a single puzzle we are tackling. When a cell reads a gene, it does not just print out final ready-to-go manual. No, it creates a rough draft that has to be heavily processed. So how in the world that raw data get turned into clear actionable data? Let's get into it. You can think of it as the message is kind of like a fragile, unfiltered stream of consciousness. To make it out of cell's headquarters, which is nucleus, and to be actually understood by the workers on the factory, it needed to be protected, edited for the clarity, and given a special tag. These three edits are extremely important for its survival and success. Okay, let's get a little technical here for a moment because it is important. This first draft is called pre-messenger RNA or just mRNA for short. It is the very first copy made directly from the DNA blueprint and it is in its raw form. It is just incredibly vulnerable right now. In the complex cells like ours, the creation of pre-mRNA happens inside the protected vault, which is nucleus. But its final destination is where proteins are actually built, in the cytoplasm. It is really dangerous journey and without some serious armor or a shield, message carried by pre-mRNA gets shredded by enzymes before it's arrived. So how does cell protect this precious message? Well, the very first line of defense is a protective cap. As soon as our message starts being created before it's even fully printed out, the cells just attach the helmet right on the front end, 5 cap. The 5 end of the RNA strand. It is not just any ordinary helmet, it is a chemically modified building block called 7-methylguanosine cap. And it gets attached to the very beginning or what we call the 5' end of the RNA strand. And this happens instantly as the first 20 or so building blocks pop out. That's what called proactive protection. And this shows how cap has dual function. On one hand, it's a shield, protects message from being attacked by cellular enzymes. But it also acts as a signal for ribosomes that are protein-making machine. When ribosomes detect the cap, it tells where exactly attach and begin the work. So now our message is protected. It's time for heavy edit. Because raw transcript is full of junk, gibberish that needs to be cut out for the final instructions to make sense. So the message is made of two kinds of segments. The good part that contains the actual instructions called exons. Pretty easy to remember because they are eventually expressed. The junk in between called introns. They are the intervening sequences. Just imagine you try to read a recipe in which every other word is totally random and makes no sense. You have to cross out, remove all that to figure out what actually to do. That's exactly what cell does next. And the editor for this job is magnificent molecular machine called spliceosome. Special small RNAs which team up to form what scientists affectionately called SNRPs or SNRPs. These are precision tools that carry out this molecular surgery. This code really gets to the heart of it. Splicing has to be perfectly precise. If spliceosome is off by just one single letter nucleotide, the entire way the message is read gets shifted and finally proteins will be totally useless. So how does splicing works? Well, spliceosomes is actually smart. It recognizes specific nucleotide sequence to mark the beginning that are guanine and uracil, GU and AG, adenine and guanine, sequence at the start and end of the intron. The introns are then cleaved and looped into cowboy's lariat. Snip it out and perfectly stitch the two axons on the other side together. That intron's lariat is released and recycled. For the final touch, adding tail to the end. Once the message has been cut at the proper end, an enzyme snoops in, add a tail of one kind of nucleotide, adenine, just A over and over again. This is called poly-A tail. Now, for decades, the textbook number for a freshly made mRNA in animal cells was a tail of about 200 adenines. A long, healthy tail is a good thing. The crucial point is that the long tail was seen as multi-tool. It acts as a buffer protecting the message from degrading enzymes and helps to kick protein synthesis into the cytoplasm. The logic seemed very simple. Longer tail equals more protein and more protection. But is it really that simple? Is longer poly-A tail always better? Well, the recent study showed something else. This quote from 2017 completely changed the script. Instead of most active, highly translated mRNAs having longer tails, researchers found out totally opposite. The messages that are actually read again and again, they have shorter tails. This is the visualization of the discovery. If you look at the average tail length for the genes that are constantly being turned on, like the ones making our proteins, they have shorter tail. But the genes that are used less often, such as regulatory proteins, have much longer poly-A tail. It suggests that active mRNAs are pruned down for the optimal shorter length for peak efficiency. So with the cap on, the splicing done and the poly-A tail added, our raw draft finally became a matured, mission-driven mRNA. So let's just recap the complete makeover. First of all, it gets a protective 5 cap that actually works as a signal. It undergoes extremely precise splicing to remove non-coding regions, the introns, and finally gets a poly-A tail that most active mRNAs for precise working have shorter length. Fully equipped and perfectly edited mRNA, cleared for launch, exported out of the nucleus into the cytoplasm, totally ready to find ribosomes and deliver precise instructions for binding proteins. Just think about it for a second. This incredible makeover is not happening to one message, happening to billions upon billions of times every single second inside you.